Myself, G. Mallanagoud, Assistant Professor of Zoology, Veerashaiva College, Ballari. Our college is affiliated to Vijayanagara Sri Krishna Devaraya University, Ballari. Today, I am going to discuss regarding the enzymes. Let us start with the introduction to the enzymes. The term enzyme is derived from Greek language. It means in yeast. Louis Pasteur was first to demonstrate that yeast can ferment glucose without being used up or destroyed. The catalytic agent in yeast was identified and was called ferment, and this process was called fermentation. Enzymes were discovered by the German chemist Edward Buchner. Name was originally coined by Kuhne in the year 1878. In the year 1835, John Jacob Berzelius, a Swedish chemist, first studied the true nature of enzymes. After 1926, James V. Sumner of Cornell University discovered the first enzymes which was obtained in pure form. Now, I would like to give the definition of an enzyme. Enzyme is a specialized protein produced within an organism which is capable of catalyzing a specific chemical reaction. The substance on which an enzyme acts is called a substrate. The molecular combination resulting from enzyme activity is called normal product. Normally, an enzyme is named by attaching the suffix called ase to the name of the substrate on which it acts. For example, maltase is an enzyme which acts on maltose. Lipase is an enzyme which acts on lipids. Protease, it is a proteolytic enzyme which acts on proteins, and so on. The suffix lytic is used to denote enzyme activity on classes of substrates such as proteins, that is proteolytic, lipids, lipolytic, carbohydrates, amylolytic or carbohydrases, and so on. All the known enzymes are proteins, hence they are mainly made up of the chain of amino acids linked together by peptide bonds. We have to study the properties of enzymes. The enzymes are having so many properties like catalytic activity, enzyme specificity, thermosensitivity, pH sensitivity, reversibility of enzyme reaction, and team activity. Let us discuss one by one. First one is the catalytic activity. Enzyme catalysis is the increasing in the rate of chemical reaction by the active site of a protein. The catalytic activity of enzyme involves the binding of the substrates to form an enzyme substrate complex. Second one is the enzyme specificity. Enzymes are specific in the reactions they catalyze. Normally, a given enzyme will catalyze only one reaction or a type of reaction. In other words, an enzyme which acts on a certain substrate is unable to act on any other substrate. However, the degree of specificity varies from one enzyme to another. Third one is the thermosensitivity. Enzymes are sensitive to temperature changes. For several enzymes, it lies between 35 degree to 45 degree centigrade at low temperatures. The enzyme is ineffective. At very high temperatures, that is above 65 degree centigrade, they are destroyed. Fourth one, pH sensitivity. Enzymes are highly sensitive to pH changes. They are active between 6 to 7.5 pH. Fifth one is reversibility of enzyme reaction. Enzymes which catalyze the reactions in both way, that is, it may catalyze the splitting of one molecule into two or unite to two molecules into one. The last one is the team activity. Enzymes usually work in coordination with each other, that is, in teams. Now we will discuss about the different kinds of enzymes. And the endoenzymes are also known as intracellular enzymes. they carry their metabolic reactions within a cell. Next one is the exoenzymes. It is also called as extracellular enzymes. They carry their metabolic reactions outside the cell. Next one is the apoenzyme. It is a protein part of an enzyme. Next one is the cofactor. The cofactor is a non-protein component of an enzyme. Next one is activators. Often enzymes are produced in cells as in inactive forms. It is also known as zymogens without enzymatic properties. For example, the gastric glands secretes inactive enzyme pepsinogen, which is converted into active pepsin by the hydrochloric acid in the stomach. Similarly, the pancreas secretes an inactive zymogen called trypsinogen, which is converted into active trypsin by the action of the enterokinase enzyme present in the intestinal juice or succus entericus. Such substances which activate the zymogens are called activators. Next one is the holoenzyme. The enzyme molecule consisting of protein and prosthetic group is known as holoenzyme. Cofactors are the enzymes may vary from simple inorganic ions to complex organic molecules and may either remain unchanged at the end of the reaction or be regenerated by a later process. There are three types of cofactors, namely inorganic ions, prosthetic groups, and coenzymes. First one is the inorganic ion. And they are responsible for the formation of enzyme substrate activity complex and the increased rate of the reactions for a specific enzyme. Next one is the prosthetic groups, non-protein organic molecules which help in catalytic activity and are found always closely bound to enzyme. The next one is the coenzymes. And they are non-protein organic molecules or carrier molecules which functions along with a particular enzyme. They, unlike a prosthetic group, do not remain attached to the enzyme between reactions. All coenzymes are derived from the vitamins. Now, I would like to discuss regarding the classification of enzymes. First one is the oxidoreductases. They catalyze addition or removal of hydrogen or oxygen to form a substrate. Second one is hydrolases. These are the enzymes that cleave complex molecules into simple molecules by the addition of water and breaking hydrogen bonds. Hydrolases are further classified into peptidases, glycosidase, esterase, etc. Third one is the transferases. These are concerned with the transfer of specific group of atoms from one molecule to another. Fourth one is the lysases. Catalyze breaking of covalent bonds and therefore degrade the substrate. Next one is the ligases. Ligases are the enzymes which catalyze the union of two molecules to synthesize a large molecule. The last one is the isomerases. Isomerases are the enzymes which bring about intramolecular rearrangement of atoms in the substrate. Now, I will take the factors affecting the enzyme's activity. There are so many factors which influence the enzyme activity, such as follows. First one is the enzyme concentration. In enzyme system, if there is an excess of enzyme, the amount of substrate transformed increases in a linear manner. But if there is an excess of substrate, the amount transformed decreases rapidly with time. Second one is the substrate concentration. Substrate concentration is one of the main factors affecting enzyme activity. The substance on which the enzyme acts is called substrate. If other conditions are constant, an increase in substrate concentration increases the rate of the enzyme catalyzed reaction. But when it is reached to a maximum, further increasing in the substrate concentration produces no additional effect. This is due to the fact that the rate of formation of an enzyme substrate complex becomes limited by the enzyme concentration. Next factor is hydrogen ion concentration. Enzymes are sensitive to changes in pH, changes in the acidity and alkalinity of the reaction medium. Every enzyme has its own range of pH in which it functions most efficiently. The excessive acidity or alkalinity renders them inactive. On the other hand, certain digestive enzymes prefer a distinctly acidic or alkaline medium. Thus, the protein digesting enzyme pepsin functions only in an acid medium at a pH between 1.2 to 2 and works optimally at pH 2. Accordingly, pepsin enzyme is found in the stomach where the conditions are markedly acidic. On the other hand, trypsin secreted by the intestine functions in an alkaline medium at about 8.5 pH. But majority of the intracellular enzymes have an optimum pH near neutrally and will not work at all in an acid or an alkaline medium. Factor. The temperature is also one of the important factor that influence the enzymatic activity. That is, enzymes are inactivated by the excessive of heat. The inactivation is irreversible. The rate of enzyme activity increases smoothly up to about 40 degrees centigrade and beyond 50 degrees centigrade. The rate begins to fall off and at about 60 degrees centigrade, reactions ceases altogether. However, there are certain enzymes which continue to act even at high temperatures. For most of the animal enzymes, the optimum temperature lies between 37 degrees centigrade to 45 degrees centigrade. Then fifth factor, enzyme inhibitors. Certain compounds like arsenic and other metals such as mercury and silver combine with an enzyme but do not serve as substrates. Thus, they block the catalytic activity of the enzyme and functions as inhibitors. The inhibitors usually resemble the substrate closely in structure. The enzyme and the inhibitor form an enzyme inhibitor complex which is inactive. The next one is the light. The light is also one of the important factors that influence on enzyme activity. In general, enzymes are inactivated by strong light. Ultraviolet light is very effective in destroying enzyme activity. X-rays may or may not be destroy enzymes depending on the circumstances. Now, we will discuss about the mechanism of enzyme action. There are two hypotheses or theories to explain the mode or mechanism of enzyme action. First one is the lock and key hypothesis. Second one is the induced fit theory. Let us discuss about one by one. First one is the lock and key theory. It was first suggested by Emil Fischer in the year 1898. According to him, enzymes have a particular shape into which the substrate fits exactly, and this is referred to as the lock and key hypothesis. Here, the substrate is supposed to be like a key whose shape is complementary to the enzyme or lock. The specific substrate molecules are found to a specific region of the enzyme molecule called active site. Enzyme reacts with the substrate molecule to form an immediate complex. This complex in turn will react to release the enzyme and yields the production of the reaction. The reaction of the lock and key theory. Enzyme plus substrate that leads to enzyme substrate complex. This hypothesis assumes that a particular lock or substrate can be opened by a particular key, that is enzyme, specially designed to open it. The notched portion of the key is equivalent to the active site since the reaction or unlocking of the lock takes place here. The lock and key model accounts for enzyme specificity. This image shows lock and key hypothesis to explain the mechanism of enzyme action. Second one is the induced fit theory of Coastland. This theory was proposed by Coastland in the year 1959. He was suggested a modification to the lock and key model known as the induced fit hypothesis. According to him, Enzymes and their active sites are flexible structures that mold themselves into a precise shape of the substrate, which enables the enzyme to perform its catalytic function most effectively. This theory suggests that an enzyme undergoes a change in its structure when it reacts with a substrate molecule to form an activated complex, and the enzyme regains its original structure after the reaction. Now we have discussed about the clinical significance of enzymes. First one, they are vital for life and serve a wide range of functions. It is shown that many diseases associated with many components of the enzyme metabolism system are now widely applied in clinical examinations as special markers for diseases. Second one, enzymes are organic catalyzers which aid in facilitating chemical reactions in the body. Enzymes are needed for the metabolic pathways in the body, such as respiration, digestion, and other important life processes. Third one is the enzymes are used in the chemical industry and other industrial applications when extremely specific catalysts are required. Fourth one, enzymes in general are limited in the number of reactions they have evolved to catalyze and also by their lack of stability in organic solvents and at high temperatures. Fifth one, enzymes are very efficient catalysts for biochemical reactions. They speed up reactions by providing an alternative reaction pathway of lower activation energy. Sixth one, like all catalysts, the enzymes take part in the reaction. That is how they provide an alternative reaction pathway. Next one is the enzymes increasing in the rate of reaction. Most cellular reactions occur about a million times faster than they would in the absence of an enzyme. Protease enzyme used in the manufacture of baby foods to pre-digest proteins. Lipase is an enzyme. It can be used in conjunction with the protease in biological detergents in order to break down and digest the substances in stains into smaller and more water-soluble vitamins. Enzymes are sometimes used as medicines to replace enzyme deficiencies in the patients, like in the use of blood clotting factors to treat hemophilia disease. Enzymes play a crucial role in the metabolic activities of all the living organisms, whether the humans, animals, plants, or microorganisms, and are widely applied in microbiotechnology and their diagnosis process. Enzymes are responsible for the catalysis of almost all biochemical reactions. The last one is, for a matter of fact, the human body would not exist without enzymes. By this, I am completing the enzymes. I hope all of you understand the detailed account of the enzymes. Thank you.